All right. Well, let's get started. Welcome, everyone. Good afternoon, good morning, wherever you are located. We are glad to have you here on today's science session topic of emerging contaminants in organics. Before we get started, let's cover a little bit of opening information here, just in terms of what we're doing here, why we're here. If you are trying to get your sense for what the pulse is of waste science, we hope to be that stethoscope for you. EREF as an organization exists to advance the science of waste and to provide that to you in a variety of ways, one of which is our science sessions, which are structured to be short, impactful nuggets of information to share where the science is at on a variety of waste-related topics. For those of you who have joined us in previous science sessions, you will know that we have been on a bit of a run related to PFAS-related content. This particular science session today will include PFAS, but we're going to branch out a little bit, talk a little bit about emerging contaminants beyond PFAS as well. These science sessions we're going to be here for about 45 minutes, maybe a little longer, but we'll definitely wrap up within the hour so that you all can go on your way, hopefully feeling a little bit more educated about what's going on with waste science. For next time, this is a great session to have today because the one that we will have after this is going to dive in a little bit deeper. We'll be joined by Dr. Yoon-Chong Choi with Purdue University to talk more specifically about PFAS in compost. Couldn't go any further without saying thank you to our session sponsors for the science sessions. These are a lot of great organizations that have chosen to support EREF in this particular way, but we have room for you as well. If you are interested in sponsoring the science sessions, please reach out to us, let us know. Your sponsorship comes with a generous helping of free registrations for the sessions, and we'd love to see your logo on here as well. A few housekeeping items. Everybody's going to be muted. We ask that if you have questions, you go ahead and submit them through the system via the chat function, via the question function, so that we can see that, and we will do our best to get to those questions at the end of the session. So let's go ahead and get started then. Let me introduce our speaker today. We are joined by Dr. MacRae, who is an associate professor in the Department of Civil and Environmental Engineering at the University of Maine. In addition to teaching environmental engineering topics, she also focuses on research in environmental microbiology, and she's part of the overall multidisciplinary materials management research group at the Maine Center and the Maine Center for Genetics in the Environment. So she's been doing a lot of work on food waste, in part generally through the research she's doing, but also through some financial support from EREF as part of a grant that she recently wrapped up. So with that, Jean, why don't we get started? Give us a little bit of overview on the importance of food waste, what's being told about what should happen with food waste and where we should go from there.
Right, thanks, Brian. Well, yeah, so food waste is a really big problem. It's a multi-billion-dollar waste issue, and it is something that we really need to be keeping our eyes on. The EPA has put out a food recovery hierarchy that we really should be following, where the focus should remain on source reduction, so not producing food that we're not actually going to be able to eat, then feeding hungry people and feeding animals. The issue, of course, in any big system like ours is that you don't necessarily have food that is fit for human consumption or even animal consumption at some point, and there's always material that just flat out isn't really food anymore. It's the discard parts. And so when I'm thinking about food waste recovery, I'm really thinking about a little bit farther down the line when we're talking about anaerobic digestion and composting. There are a few fancier ways of recovering value from food waste, but those are the two major outcomes of food waste recovery.
Okay, so to your point, when you're talking about, you're referring to the hierarchy here, we're really referring to these uses after feeding people, after reducing sources, after feeding people, and after feeding animals. It's really those sort of bottom three parts of the hierarchy. Is that right?
Well, yeah. So for the recovery part, I guess I'd put it above landfill and incineration. So so composting and anaerobic digestion are really the area that I'm focused on at this point. Certainly being higher up in the hierarchy is really important, but there's definitely aspects that you can't really do. There are materials that can't be recovered for human consumption, and things spoil sometimes, or animals get into it, so you just flat out have to have other alternatives. And so composting and anaerobic digestion are the ones that primarily we think about.
Okay, so just helping us out here in terms of from the policy perspective, because certainly food waste is one of those big topics of conversation about when we speak about waste diversion. Where should this material go? A lot of this doesn't happen in a vacuum. I mean, from an operational standpoint, there's the policy landscape that needs to be navigated. Can you give us a little bit of background on that policy landscape for food waste recovery as it relates to some of the work that you've done?
Yeah, so I think across the country, states are trying to move toward a more sustainable model, and they're trying to divert more food waste. And there are a variety of models that have been tried in different parts of the country. I think Connecticut might have been the first one to try sort of large scale bans, so that if you're a producer of food waste above a certain amount, then you can't send it to landfill. You have to come up with an alternative. And California then implemented graduated source separation requirements for large and then progressively smaller generators, which is the same approach that's been taken by Massachusetts. And Vermont is kind of an interesting case in that they've gone so far as to require that residential customers divert their food waste. So everyone in the state needs to do it in Vermont. So there are a variety of different ways that people have tried. So there's in some cases you only have to divert the food waste if there's a composting facility or an anaerobic digester within a certain distance of the institution or the town, and so that deals with some of the logistical problems of getting the waste from where it's generated to where it needs to go to be treated. But I think having some form of regulation and a requirement is really helpful for people who are thinking of setting up a composting facility or an anaerobic digester, in that it enables them to plan for a certain amount of waste. So even though data in solid waste is terrible, some data is better than none, which is what you get if there's no mandate, really. So I mean, you can reach out to particular potential customers and try to generate business in that way, but it's much easier to plan when you know that there's going to be a certain amount of waste generated, and that's a possible feedstock for you. So particularly in anaerobic digestion, where there's a really big upfront investment that's required to set up a system like that, and it's a little bit less scalable than composting operations, so you can't sort of gradually move up. It's really important to know that you're going to have a reliable source of material to work with. So there's a lot of social and economic issues, I think, that sort of go into coming up with a plan and then having a system that works throughout a state or a region. So my state of Maine has got nothing. Like you can see, we're gray in the upper right corner of that map, and we don't have any requirements. And partly it's because the legislators are afraid that it's going to cost a lot of political capital for them to put a ban in place, and then the communities and municipalities that currently govern how waste is managed in the state, if they don't like it, then the legislators are going to hear about it. So so I think it's good to have some form of regulation to make it easier for an industry to grow up around that, but there are some roadblocks there, and there's resistance in a lot of places.
Mm-hmm. Yeah, and you mentioned this in your comments earlier. In addition to the policy landscape, which can create challenges or help be an impetus to moving the organics diversion equation further along, you mentioned the logistical aspects of things, and certainly that's something that we've seen in some of the internal work that EREF has done when it comes to availability or access to composting facilities. That can be challenging in certain areas, and sometimes, in some cases, based on some of the policy work that we've done internally, we've seen, for example, that the policies are a little bit out ahead of where the infrastructure is. In other words, if you were to divert the organics from, say, a landfill setting, there really isn't enough capacity for some of these facilities to handle that, which is one challenge. The other challenge is some of the work that you've been doing along the lines of risks to the diversion of organics, and I'm curious to have you touch on that. When we talk about risks, I would imagine most folks would think of food waste as a feedstock for composting, and when they think of the word risk, it really relates to the traditional contaminant that many facilities sort of traditionally face, like glass or shards of plastic, or things like that just traditionally don't compost, or that cause the quality of that compost to go downhill. But some of the work that you've done really has gone well beyond that. I mean, we're talking about different kinds of risks. So maybe you can touch on that element of risk beyond the traditional contamination there, and give us a little insight to what your focus has been.
Sure. So on the next slide, I think I drew this little figure of agricultural production going to processing and packaging, to distribution and resale, to consumers, and then to waste management. And there's a waste arrow in all of those places. There are different kinds of risks. And so as part of the research that we did survey people who are working in the field of food waste management, and they really were focused on physical properties of the materials coming in. So they were most concerned about plastics getting in, and the potential for glass and sharps causing, a reduction in the quality of their material, and sometimes not even being able to get rid of it. So that was definitely viewed as the primary problem for them. However, we did get a few little inklings of interest in the presence of pathogens in waste materials. So I'm not really quite sure where the comment came from, but one of our respondents was, thought that there was potential for liability there, if pathogens made it through. Although I think the risk of that is relatively low, given how most of these operations are well run and they reach a high temperature. But there was less concern and interest in trace contaminants like heavy metals and trace organic contaminants that can get into our systems. So we were thinking about it in a couple of different ways. One is just that, food waste should be low in these things, because hopefully our food is low in those things. It's a little bit different from the biosolids landscape, where, wastewater is where we try to get rid of all of the stuff that we don't want, but food is what we put in us. And so scraps and parts of that hopefully are relatively low in these things. But we wanted to actually see what the levels were, to make sure that the things that potentially could build up in a system are not building up to a degree where we're creating problems for ourselves by trying to circularize the system to a greater degree. So the more food waste we divert, the more material winds up going back onto agricultural lands, the more opportunity there is for amplification of chemical contaminants that can grow over time. So they're constantly being produced, but they're not degrading at the same rate that they're being put onto the land. So that's the issue in terms of amplification. Biological risks that we were thinking of. So I did mention the pathogens, but antibiotic resistance genes are an issue in food systems, because we use antibiotics, or up until very recently, they've been pretty much unregulated use of antibiotics as growth promotants in livestock rearing. In some cases, the antibiotics that were being used are ones that are also used for human therapy. So the development of resistance over time has increased. You can sort of see a parallel increase in antibiotic resistance in hospitals, which means that there's some form of connection between the use of antibiotics in agriculture and the presence of resistance in the environment. And there's a school of thought that there's like this pool of antibiotic resistant genes that are in the environment that potentially can move into pathogens or wind up in our microbiome, which, inside our guts. And once they're there, if you're being treated with antibiotics, there may be a mechanism for the microorganisms there to resist therapy, which is a bad outcome if you're sick. So, so those were the risks that we were looking at, as well as, thinking about the economic and social impacts of putting a lot of investment into recycling of food wastes and trying to get people to think about doing it properly so that we can reduce the risk to the overall system.
Okay, so, and that's interesting because you mentioned the antibiotic resistance genes, which kind of brings me to the fact, how do they get in compost in the first place? And along those lines, how do these other risks, as you've categorized them, physical, chemical, biological, how do they get into the broader spectrum of organic waste in terms of sources? Where are these things coming from, across the spectrum here?
Right. So they can come from a lot of different places. So here, if you looking at the agricultural production side of things, the pesticides, fertilizers, and antibiotics are used in agriculture. Pathogens are generated with livestock, and that also the use of manure gets pathogens and just fecal material onto the plant materials as they're harvested. They're not sterile or pristine. They do get fecal material on them, which can contain pathogens. And trace contaminants really primarily come from the air, water, and soil that the food is grown in. So all of our systems are connected, and so the closer you are to an industrial area, the more likely you are to have some airborne contamination coming down on the soil, which over time will tend to contaminate that. And irrigation water is a source of contaminants as well. So these things do get taken up into the plants. And so if we're thinking about maintaining a system that is safe or safer, we want to make sure that there's as little of that as possible escaping into the environment to actually get into the food in the first place. So there's plenty of routes for contamination. In this country, we're, in a lot of places, the irrigation water is good, and the water quality and air quality are not too bad. And most of our food is being produced in places where there isn't gross contamination like there are in abroad. But there is some contamination that is coming from those routes. When you get to processing it, yeah, sorry.
Oh, no, go ahead.
Okay, when you get to processing and packaging, there are additives that are incorporated and packaging materials. There have been some very interesting research recently showing that packaging materials that are made out of recycled plastics have higher levels of metals, for example, and there's a whole bunch of other kind of oddball things that have been cropping up in the literature recently. And maybe that's sort of partly due to the disarray effect, from China coming out of the recycling ecosystem. It's hard to say where all of that stuff is coming from. Having to deal with new situations as far as recycling is concerned. Pathogens can be added into the system just by poor storage or handling or unsanitary conditions where things are being processed. At distribution and retail, you can also have unsanitary conditions. You can have contamination with cleaners while you're trying to maintain sanitary conditions. And different packaging materials may be incorporated. And for packaging materials, some of them leach, some of them have PFAS associated with them. And so there's a variety of ways that contaminants can be introduced at that stage. And then finally, at the consumer level, households and institutions operate differently, and people, there's different levels of understanding and care and attention taken to separating wastes. So that can introduce just trash into a system, or wastes that just don't belong with the food wastes, including packaging, pathogens, pet wastes, and so on. So there's a lot of places where things can actually get into the system. And when we get to the waste management side of things, some of the contamination can be removed. So pathogens should be killed off if the system is well run. Organic materials are in some cases biodegradable, and they may degrade in the system, or they may get to be a higher concentration because the background material is being degraded. Some contaminants are completely unaffected, and some may increase if there's if it's a breakdown product from some larger organic contaminant. So a lot of potential places for stuff to get into the system. And, you know, luckily we still have a good and a safe food system. So the concentrations here should be low relative to what we're used to seeing with biosolids. But on the other hand, we want to really be sure that we're not introducing things that are going to amplify.
Right, well, and I feel like the timing couldn't be any better, because with the policy conversation, with the focus on diversion, as we've had that focus now for a number of years, but it continues to grow. The call, I think, publicly continues to grow for folks to the pressure's building to continue to divert organics. So this kind of effort is critically important. If in particular more organics are going to be shifted into these different endpoints, it's helpful to understand that. I realize that some may view this kind of work as Gene, you're the harbinger of sorrow in some respects, saying here are these different risks, but without understanding what they are, it's going to be very difficult to control for them. I think the PFAS conversation is a great example of a compound that's been around for a long, long time, and really only just now we're getting a handle that there are potentially some significant risks posed in the PFAS equation, and in other cases not potentially. There are absolutely some risks. One thing that I find really compelling is in your bio, it's noted that you not only are a researcher, but you are a science educator, a teacher. You're teaching courses. And one of the things that I love about that is that there's the intersection between the research that you're doing and educating folks on exactly how science is done. And, you know, with my own experience in research, I know that really the research is only as good as the methodology that's used. And I think that's lost on a lot of folks who aren't in the research space, that a tremendous amount of time goes into creating a methodology to actually conduct the research, so that at the end, the results are results that you can deem valid. And so tell us a little bit about how you went about this research, and how you set it up, and why you selected some of the things that you selected to test for.
Okay, yeah, so we had a couple of aims in doing this research. One of the questions we really wanted to know was whether, if you told people that they had to separate their food waste, would they do a worse job than if they were doing it voluntarily? So we did a bunch of meetings with people all around the state, and there was some really clear opinions that, you know, we cannot tell people to do this, because then it's going to affect the quality of the materials that we're able to collect, and then we're going to end up with a contaminated system. So we were concerned about that, and we also wanted to look at the impact of source types to find out whether some sources were worse or better than others, because again, that was a perception among the people that we were talking to, that you know, one group would say, don't ever go collect at schools, and another company would say our worst are such and such. So so we wanted to have a look at different available source types. So we basically sent graduate students to go dumpster diving and collecting source separated material at individual collection sites. We removed the physical contaminants, weighed them, cataloged everything that was in there. We then processed the food waste in a food processor, and we tested for heavy metals, halogenated organics using a bulk method called EOX, or extractable organohalides, so that we didn't have to prejudge what might be in there. And we took a subset of samples and looked for PFAS in those. We looked for several important foodborne pathogens because we figured those were most likely to be present in the system. And we looked for antibiotic resistance genes to antibiotics that are used in agriculture. And we also did a little bit of high throughput sequencing to see if there were additional pathogens that we were missing using those methods. So our goal was to figure out whether regulation would impact the degree of contamination on any of these things, any of these parameters, and we were looking for physical, chemical, and biological contamination of the wastes.
So in the selection of these different categories, and I'm glad you clarified halogenated organics. I thought that was a breath mint you'd get at GNC. But as far as these categories, are these all regulated categories, or are some of them, I know some of them are, but a few of them may not be, and maybe give us a little bit of insight on why you selected some of these that may not be regulated at this point in time. Is it because you consider them, some of the higher risk emerging compounds out there?
Yeah, so halogenated organics are regulated. Some of them are regulated. So some individual compounds are regulated, and we figured if we could see some halogenated organics using the bulk method, that would indicate that there's something in there and it's at a relatively high detection limit. Worldwide, though, there's only one place that requires this sort of bulk measurement, and that's in Europe. There are places where biosolid application is limited by adsorbable organohalogens. So that's same method, but used for liquid samples. The PFAS is beginning to be regulated, but it wasn't certainly at the time when we proposed this work, and it's been on my radar screen for a long time because I find them terrifying because they're impossible to break down. That the carbon fluorine bond is so strong that that it is really, really hard to break these compounds down. So once we make them and release them into the world, they're kind of out there and they find their place based on their chemical properties and who's unlucky enough to bump into them. So I wanted to include those ones in there. Antibiotic resistance genes was also of interest because of the sort of increasing global problem with antibiotic resistance and the fact that they've been used at very high quantities in agriculture. Like 2/3 of the antibiotics that are used in the States, at least as of a few years ago, were used in agriculture, not in people. So, we use these for way more for growing our food than we do to keep ourselves from, healthy. So, so that's why that one is included in there. I really felt like we didn't have any data, and it would be important to actually have established some of the data on these things so that we can figure out if it's growing or changing over time. So I feel strongly that we need to be keeping an eye on the system, and we do need to have some data on things that we intellectually it makes sense that this would be a problem, but there just really is not that much data available in the literature. So that's why that was included.
Okay, great. Well, speaking of data, then let's get right into what you all found as part of this research. Walk us through some of that. Some of the information that you all were able to pull together from the research.
Sure. So first I should say that we really didn't see any difference in regulatory environment. So, I mean, that was kind of interesting, just flat out, it didn't make much difference. And I think that's encouraging because it means that regulators don't have to be quite as afraid as they are about proposing diversion requirements. So that'd be good. Differences by source, we didn't see much either, but you can actually see from these box plots. So the way to read these is the heavy middle line inside the box is the median value, and the box itself covers the 25 to 75th percentile, and the vertical lines show the range of data. And there's a fair bit of variability on all of the everything that we measured. We got a fair bit of variability. The only differences that we saw were the ones that you can see here by source. So the hospital copper concentrations were a little bit lower than the residential copper concentrations in the food waste. And in tetracycline, the grocery was a little bit lower than hospital. So the copper concentrations, all the values are low, and I think that this is just, variation among samples. It's probably not something to be worried about. Any of these values are below any regulatory level. That was true for all of the heavy metals that we measured. Most of them were not detectable, and using this EPA method, and we weren't concerned about the heavy metal concentrations. The tetracycline resistance genes being higher in the hospital than the grocery store waste was, marginally interesting, because hospitals are known to be hotspots for antibiotic resistance because they use a fair bit in hospitals. But again, it's all sort of within the variability of all the rest of the samples. So we don't necessarily think that that's a particularly real difference, but we were interested, and we thought it was important to actually have the order of magnitude of the abundance of these genes and the relative abundance of the genes. So in this case, you can see those middle lines sort of cluster around the -3 log, which means that about 1 in 1,000 organisms has antibiotic resistance genes in it. And that's a fairly high proportion of the organisms.
Now one thing about the way that this is expressed, and not getting in the weeds too much, but is this plot measuring kind of the level of metabolic activity, or really just more of the presence of the genes? And I know that's a distinction between whether they're expressing this, in other words, actively manipulating this gene, and it's being used by the microbe, or is it really just that it's present in the community?
It's just that it's present. And I think I'm going to be talking a little bit more about the antibiotic resistance genes shortly, and so I can say it now or I can say it later. But the thing about antibiotic resistance genes is they tend to be carried on these mobile genetic elements, which are sort of transportable pieces of DNA that can be shared between one microbe and another, which is in part why antibiotic resistance genes move around so much, and they can actually be shared between microbes that are not very closely related to each other. So, so that is why we're concerned about the general level of antibiotic resistance genes in the environment, even if they're not in pathogens. So one of the reasons for looking at pathogens and antibiotic resistance genes together was to know if there's possibility for crossover into the pathogens, but actually just the presence of the genes themselves is enough to be concerned about.
Right. Okay, thank you for that background. So what about some of the other things that you all looked at as part of the study?
So the halogenated organics, we looked at these because they're synthetic chemicals for the most part. They tend to accumulate because halogens are not normally associated with organic matter, and so there aren't normal and common biodegradation pathways to get rid of them. So once you produce them and you put them out into the environment, they have a tendency to accumulate, and in many cases they're also hydrophobic, so they build up in tissues as you move up the food chain. So there are all kinds of examples of halogenated organics that have been banned 30 years after they came out onto the market, and now we're seeing some that are looking like 70 years after they came onto the market they might see bans because they accumulate, and it's not until many years later that you discover that there's some awful health impact of these long-term low-level exposures, and the opportunity for those things to grow in the environment produces a dangerous situation for wildlife and humans. So we wanted to know if, to the extent that they were out there in food waste, as opposed to, sort of in comparison to biosolids where there's a little bit more data. And we were unhappy to detect any, but it was only 14% of our samples had detectable amounts. It looks like there's one that's super high compared to the others, but it's still below the regulatory limit for biosolids application in Europe. So relatively low concentrations here, but something to keep an eye on just because of the overall potential for problems with this type of contaminant.
Gotcha. And then, yeah, and for broader antibiotic resistance, you guys also looked at penicillin, is that right?
Yeah, so we were looking at penicillin and tetracycline resistance genes, and they had similar abundances overall. The overall concentrations that we saw of these contaminants were a little bit higher than you would expect to see in a normal agricultural soil. So, so they're a little bit enriched in the food waste compared to what you would see in typical soils. But there's not a lot of information out there on these abundances so far. Like, I think we're going to end up with a bit more data, because certainly people are taking an interest in antibiotic resistance in parts of the world where use of antibiotics is even less controlled than here. We're going in the right direction by imposing a little bit more limits on the use of antibiotics, and other parts of the world have really not restrained or reserved the use of antibiotics to human therapeutics. The other antibiotic that we were looking at was colistin, which is the last resort antibiotic that's used when you're faced with an infection that is multiply resistant. So you've tried this, you've tried that, and so at that point you go to colistin. And unfortunately, there are brand new, as in 3 years ago or 4 years ago, genes that have been discovered that confer resistance to those antibiotics. So luckily in the US, we haven't been using colistin in agriculture, which is good. It's been protecting us. So we didn't see any colistin resistance in the samples that we looked at, and that is sort of backed up by a project that was done in California, where they were looking for it in 1,000 samples, and they didn't see it there. But it has been seen in people in the US, so it's something that we should, continue to be looking for.
Okay. Well, and what about today's conversation? The elephant in the room. What about PFAS?
Yeah. So we did detect PFBA, which is one of the small PFAS compounds. So PFAS are perfluoroalkyl substances, and meaning per and polyfluoroalkyl substances. So meaning that they generally have a hydrocarbon tail that is bound up with fluorine, and at the end there may be a carbonic acid group, or there could be a sulfonate group, or another group at the end that can break down into one of those two. These compounds are super useful in that they're very stable. They don't break down. They're resistant to heat. They have a lot of really useful properties. So they were used for 50 years without apparent problem. Just, basically everybody thought if they were that stable, they wouldn't have any biological impacts, but they do. So they've been found to have pretty severe impacts, including immunological issues and a bunch of others. So we looked for PFAS in the food waste, in a subset of food waste samples that we collected, and 60% of them were positive for PFBA, which is a 4-carbon fluorinated chemical. And the concentrations are fairly low. Now on my screen, I can't see the bottom, but it's parts per billion. It's up to 1 part per billion, which is sounds super low, but it's only sort of low for these compounds. It's much lower than what you typically find in biosolids. So I'd say 1 to 3 orders of magnitude lower than the concentrations in biosolids. Biosolids get everything. It's hard to be a biosolid because you're going to be contaminated. But food waste, you would hope, would not have this. What was interesting was that we didn't see any PFOA or PFOS. So those are the two compounds that were the most widely used for a long, long period of time. And those are not in our samples, which kind of suggests that the concentrations of those now legacy chemicals has gone away from food. It used to be detectable in food, and here we're not finding it in food waste. So that part's good. The question of what it means, though, now that we're looking at a completely different compound, is a difficult one to answer. So PFBA it doesn't build up in tissue to the same degree that the larger ones do. So that should be a positive thing, but it's more mobile, so it moves into water more easily, and it's harder to treat to get it out of drinking water. So that's bad. And it's also taken up by plants to a greater extent than PFOA. So PFOA is an 8-carbon compound, PFBA is a 4-carbon compound. Otherwise, they're very similar. And PFOA could be 10 times higher in the soil and be the same concentration as the PFBA in the plant. So you get 10 times higher uptake of PFBA into the plant than of PFOA. So it's, there's a high concentration factor, which could really account for why we saw PFBA in the food, because once it's taken up into the food, well, plants can be food, and so can animals that eat the plants and so on. So,
right.
Yeah, go ahead.
Well, quick question for you: How many PFAS compounds did you test for? I may have missed that earlier. So you're showing 3 here. You said you also tested for PFOA and PFOS. Were there others that you also tested for?
Yeah, the 17 compounds. And it'll be a alphabet soup if I try to rattle them all off. But so we were looking for the small ones to the large ones, and but we weren't looking for any of the things that break down into the PFAS. So so we had 17 compounds, and these are the only ones that we saw. And every one that we got a positive in, we did detect PFBA, and we had a couple with 6 carbons. That's the PFHXS, and the PFNA is a 9-carbon compound. So it's one of the larger ones that apparently also has a higher tendency to accumulate on plants. So that's normally absorbed to the outside of the plant versus being taken up into the inside of the plant like PFBA. So why that one accumulates more than PFOA, I'm not sure, but.
I hear you. Now, this may be outside the scope. I am curious to know, though, in looking at this graph that you shared, and it almost looks like there's a bit of a grouping around the residential sources that you compiled. I don't know if you can speak to this, and forgive me if you can't, but do we know or have a sense for where the PFBA could be coming from these particular sources? Or do you want to speculate on that even?
Well, it's hard to speculate, but so I mean the sites that I know of in Maine where there's been PFAS contamination have been primarily places where biosolids were applied to land, and that has resulted in contamination of the water. So there's actually a few places, and once the water is contaminated, that's going to have a relatively high amount of PFBA, and so if it's the water, that can translate into the plant. So it can just be the area where the material was grown. I think there's probably a lot of airports, anybody who's getting drinking water or irrigation water rather near an airport or military base, quite likely those places will be contaminated with firefighting foams that are a big source of PFAS. Long-term biosolids applications of various industrial wastes. I mean, this stuff was used for such a long time. And right. The mobile portions of it really are those smaller compounds that are taken up into plants. So the contamination can be old, and now that we're starting to look at it, I'm sure we're going to find it in more places. So to me, it was interesting to have a look here in our food waste, which to me should be pristine. It's not quite pristine, but it's quite a bit better than if we were looking at biosolids. So we're talking orders of magnitude difference.
Gotcha.
So,
okay. Now, so bringing this kind of to a head, because I want to get some questions from the audience. But thinking about these as risks and the significance of these risks, can you weigh in on that a little bit and kind of summarizing that a bit? Because I heard you earlier mention that there are, of the compounds that you tested for, quite a few of them were under regulatory limits, in some cases well under regulatory limits. And so in that respect, what is your sense for the relative risk posed by some of these? And are there others that you would point to that say that these are, from a scaling that risk level, these are certainly on the higher end in terms of concern versus the lower end based on what we've seen?
Right. So I think the biggest concern to me is the PFAS contamination. So because that is much more difficult to get out of a system once it's in. Ultimately, what I think is relevant here is that we continue to try to move into a more circular food system, and that we keep our eye on the whole system. So I think the best way to deal with PFAS is to try to separate PFAS from our food. And it's going to be difficult in sites that have legacy contamination, but there's a few things that we can be doing that will keep PFAS lower. And so first off is PFAS are used in food contact packaging and papers. And if I were operating a composting system or an anaerobic digester, I would make sure that all of that material was separated out before the material goes into the composting facility. So I would be, working really hard with all my clients to make sure that they're not including paper with the materials, at least until there's better labeling and it's really clear that the materials that are called compostable don't have things that don't degrade in compost. So we don't have labeling like that at this point. So there's all kinds of stuff that gets labeled as compostable that doesn't really go anywhere. It just sort of gets smaller, but it's still, the same material. And PFAS in food contacting papers, there's a whole bunch of evidence to suggest that that ultimately gets into people as well. And so there's good reasons from a health point of view to keep them out of those contact papers. And there's good reasons from a waste management point of view to try and keep them out of waste management. So we should basically ban those things, and then everyone can go on about their business. Antibiotics use should be reserved for therapeutic uses for humans and animals. I'm not saying that there should be no use in agriculture, but I think potentially we could be using different antibiotics in those two settings. And the USDA is moving in that direction now, and that's good. They're asking veterinarians to approve any just feed uses of antibiotics. So that's a good move, and hopefully that's going to reduce the amount of antibiotic use in agriculture overall. And we really need to learn more about the fates of all of these compounds during treatment and also during pre-treatment. So there's, depackaging and so on. It's hard to say how much of the materials that are actually in the packaging material will find their way into the food slurry if you're using a depackager. I haven't seen any papers on that really. So it would be interesting to see if there's any leaching or if there's microplastic production or a variety of other issues that could potentially come up there. And really just think about the whole system. And think about, how far back you can do the work on preventing contamination in the first place, because people who are managing the waste have very little control over the materials that come in. Like someone throws it into that bin, and now it's your problem to deal with. And I think everyone would be a lot happier if the stuff coming in was clean, so that you could have a clean product at the end that you can sell and people can safely use.
Gotcha. Okay, great. Well, thanks for that. Let's move on in the remaining time we have. We've got a number of questions from the audience, and one of the first ones relates to the actually some additional potential persistent chemicals. They wanted to know if there was any data on persistent prescription drugs, whether you did any of that work, or just in terms of your literature review, if you came across any of that.
Well, a lot of prescription drugs do stay during treatment. Certainly, I guess I have less experience looking at this in composting and anaerobic digestion than in on the wastewater side of things, but they do tend to persist. And part of the design of drugs really is to make them stay active in your body for a long enough period of time, which means they don't break down, unfortunately, which is part of the problem, which is why we end up with them in our water. One of the reasons for doing EOX was in part that should capture, some of those drugs that are used in therapy and as antibiotics. They're chlorinated or they're brominated, or in order to make sure they have a long enough half-life in the body. So they're definitely a problem. Degradation is variable by compound and by chemical composition, and that also determines whether it winds up in the air versus the soil or versus the water. And unfortunately, it's a bit of a whack-a-mole game for these compounds that are not very degradable, is that we tend to, I got to get them out of the water, so I turn it into a solid waste problem, and then it goes to the composting facility, and then, it heats up so much it winds up in the air. So, we're all sort of moving these compounds around, and unless we have actually degradable materials, it's a persistent problem, and that's why we have to be thinking about this as a system.
Mm-hmm. And for these variety of compounds, so we had a question come in that asked, are all these chemicals found in the food waste or in the finished compost, or perhaps both?
So this, what we were looking at was just the food waste. So our study was purely on that, but your next speaker is going to be talking about what happens to contaminants, or at least PFAS, after composting and after processing. So that's really, that'll be exciting to look at, because that's what's relevant, because if that's the material that's going to wind up on the soils. But you do need to know what goes in order to figure out how well the system is done on the other end. So I'm hoping that we can look at some of these more important contaminants in food waste after treatment as well, and also be thinking about how the packaging and some of the pre-processing results in more or better or worse outcomes in terms of the inclusion of some of the materials that may contribute to the contamination.
Okay, and another related question in terms of did you all sample, was it mixed food waste, or did you have specific kinds of food waste that you were parsing out as part of the research?
So we were looking at just whatever was in the food waste tote at the different sources that we went to. So we went to the hospital and dove in their dumpster, and then we went to the schools and collected waste from there. So really depended, the materials were quite variable. In some cases, there was, in one case, I think somebody just dumped a giant thing of, like, maybe it was old cornmeal or something, but the whole thing was this yellow powder. I was glad it wasn't a white one when the grad students came back, but so yellow powder, and then, other ones would just be what would typically look like in your kitchen garbage. It was highly variable, and that contributes to some of the variability in the chemical contaminants that we were looking at.
Right. So someone had a question about the testing approach and how feasible is it to do testing of heavy metals and PFAS or organic samples from different point sources on a large scale. I think this is kind of getting at just the logistics of doing this, and I'm sure cost is certainly a factor, because I'm well aware that when you start testing things like PFAS, that is, even with a small number of samples, that's not a cheap proposition. So, but do you want to weigh in on that?
Yeah, so metals are not that expensive to test, but they're not really that big of a problem in food waste. So, you know, it's on a regular basis, I think actually monitoring for that would be cost effective, and it might be able to help us find issues if and as they arise, but less of a problem, I think, than PFAS. PFAS, if you send them out, it's upwards of $200 a sample. So it's not something that is probably going to happen on, you know, batch by batch, every batch at every composting facility. It's that's more money than you're going to get back, I think, in the end. So you're the idea of testing all of the materials coming in is just it's prohibitively expensive. So we're going to have some methods that come down in price. That's in part why we looked at that bulk measure of EOX instead of looking for individual pesticides and herbicides and all the rest of it, because you know, for each individual chemistry method that you have to use, which might capture 20 or 30 different contaminants, but you know, they're a group of contaminants that have similar properties. And so if you open that up and you're starting to look at you want to know pesticides and industrial solvents and herbicides and PFAS and, you know, flame retardants and the next thing, you're talking $100 to $200 for each one of those different analyses, and it's just it's too expensive to do on a regular basis until we come up with some sensor-based methods or methods that are a lot cheaper. And you know, food waste it's variable and it's difficult to work with. So coming up with a really cheap method is much harder than, you know, in the medical settings where I think detections are moving much quicker. Like the marketplace for finding new detection methods that find low levels of important contaminants of concern is moving along quickly, because there's a lot of money in medicine, and second, they have cleaner materials to start with, like spit's cleaner than food waste. It's less variable.
So you mentioned herbicides in your response. That was one question. If you did happen to look at herbicides as part of this study, it is a kind of a persistent chemical contaminant in compost. Did you guys look at that at all?
We didn't look at it separately. So we were expecting, you know, that would fall under the EOX method. And of course, now, because it's a bulk method, you can't really say what exactly was in there. But definitely that was something that we heard from our survey respondents, was that was a really important issue for them, because if you have persistent herbicides there and somebody puts it on their shrubs and their roses, they're mad at you when you've killed their roses because there was a herbicide in the finished compost. So, I mean, that's kind of the nightmare, I think, of all the folks that we were talking to about, you know, not having control over the input materials and not really having good and inexpensive and, you know, practical methods of assessing the materials before they go out the door.
Okay, great. Well, I think at the top of the hour, I do want to make one quick note. Thanks to Kerry Oceans at the U.S. Compost Accounts for this tickler, but he noted that we are reminded that all the certified compostable products, whether BPI, the Compost Manufacturing Alliance, or European, are already PFAS free. So there are things in place, but to your point, Gene, as you were noting, the state or labeling laws really can help in making sure that we have a stronger labeling component there. But it is important to kind of note that. Being at the top of the hour, I apologize to those of you we could not get to in terms of your questions. If we didn't get to you that and you really would like us to address that, please shoot us an email. We're happy to connect the dots between your question and Dr. MacRae. And we'll go ahead and wrap this session up. Thank you all so much for coming. Dr. MacRae, thank you so much for your time today and for sharing a little bit about what's going on with your research.
It's a pleasure. Thanks.
All right. Have a great day, everyone.